Our last talk is going to be by Joshua Shulman, Baylor College of Medicine, who's going to talk about the transcriptome and hopefully some drug development.
A lot of opportunities for drug development, but not yet. But maybe Jim will have some ideas based on what I'm going to talk about. So I'm really excited to be invited, and I think the meeting organizers, to share with you some of our work on the Alzheimer's disease brain transcriptome. So the human brain transcriptome, or all the expressed genes within the brain, is really can be thought of as the mediator and therefore the executioner of the genome's effect on human health and disease. But also, as we've heard, as pathologies impinge on the brain and affect brain function and health, the transcriptome is also a readout or an integrator of all those lesions and the way they affect brain cells to change their gene expression patterns. So in those two ways, the transcriptome is an important cause of changes in human health and disease, but it's also a readout of the effect of these disease processes and is therefore maybe an important biomarker of what's going on in disease. So that makes the transcriptome very interesting and exciting and important, but of course it also is the great challenge of interpreting and understanding the transcriptome, because it may be a cause, but it also may be an effect. And so what I'm going to talk to you about is how to disentangle those things. We have been a part of a large consortium effort, and I feel lucky to be a part of this effort, called the Accelerating Medicines Partnership Alzheimer's Disease Consortium. This is a public-private effort that involves National Institutes of Health and several pharmaceutical partners. The goal of the effort simply is to discover new drug targets for Alzheimer's disease. And it was a very large data generation project, and the main approach taken by 6 tightly linked academic groups that we were a part of was to characterize gene co-expression networks that are associated with Alzheimer's disease in more than 2,000 human brain autopsies. So I don't have time to go into the excruciating details of more than 5 years of work that was behind this, but a large number of brains were brought together. RNA sequencing was done across these brains at a number of centers, and efforts were made to identify groups of genes that are changing together in relationship to Alzheimer's disease diagnosis, the presence of Alzheimer's disease pathology, cognitive changes that are occurring in subjects, specific types of pathology, including amyloid plaques and tau tangles. I'm showing you one example from a recently published paper of this, and a specific example of a particular RNA co-expression network, which you can see here modestly increases, but significantly increases, its expression in the brain in relation to degree of cognitive impairment. So in the rest of the talk, I'm going to talk about 30 such co-expression modules that were identified based on a meta-analysis of this very large data set. Each of these modules is significantly associated with Alzheimer's disease. And important, these gene modules are very large. The mean size of the modules is 2,000 genes. So these describe large, vast expression networks and signatures that are changing in the brain in relation to disease. Again, that gets at part of the challenge of interpreting them, which is really what I wanted to focus on the next slide. So there's great promise in this type of data, but there are challenges. One, Alzheimer's disease is not a single insult. There are 2 pathologies that we've heard about, amyloid and tau. And of course, we've also heard in some of the talks yesterday that there are other things going on in the brain, like cerebrovascular disease and pathology of other neurodegenerative conditions, and life experience. And so how do we figure out what changes that we find in human brains are specific to Alzheimer's disease? Of course, we can only look at the brain transcriptome once after somebody has died, but Alzheimer's disease occurs over decades. So dissecting out the dynamic age-dependent changes that occur in the brain is also a great challenge. Now, if you want to study these networks or these genes, you need to pick an experimental model. And that's a challenge. What model is appropriate to study a given network? And how do you even define conservation from human to a model, like a mouse model, at a network level when you're talking about 2,000 genes that might be changing in tandem? And of course, the final point is at the end of the day, we want to figure out which changes are causal to disease, because those are going to be the ones that are druggable. Or those are the ones that we hope to drug to alter disease progression. Druggability may not be the case. Our potential solution to this is to integrate the human data with similar brain transcriptome data from a large number of mouse models of Alzheimer's disease and other neurologic conditions. And the conceit behind this approach is that what we want to do, is pinpoint correspondences between gene expression changes from human brains that are associated with Alzheimer's disease with similar changes in mouse brains that are caused by controlled experimental manipulations. That's the key difference when you're talking about a mouse model or a Drosophila fly model, is that we can experimentally control the manipulation and measure the effect on the transcriptome. Okay, so this slide is the key to understand everything that's going to follow. Now's a good time to pay attention. We developed because otherwise you're going to wake up in 2 slides, you're going to be like, what is that? We developed an approach to determine correspondences between human and mouse brain transcriptomes and to visualize the overlap and the significance of that overlap. And so what you're going to see on the following slides are a series of these grayscale heat maps. The columns represent mouse differentially expressed gene sets. So each column represents the results of transcriptome assays in a particular mouse experimental manipulation. And the rows represent these 30 consensus gene sets that I described that are associated with human Alzheimer's disease from postmortem brain tissue. One subtlety that I should mention, because it may confuse some of you, is that the co-expression modules were developed from 7 different brain regions, and the brain regions were treated independently. So what that means is that each module is not completely unique, but there's a lot of overlap, and there are clusters of modules. So you're going to see patterns of overlap that occur in groups of clustered modules. As far as the mouse data goes, the data comes from 100 or so available studies, more than 2,000 actual mouse brain samples. And from all of these studies, we extracted 378 curated experimental comparisons. From those, there are 250 experimental comparisons that actually provide informative data that you're going to see in the following analyses. And so what these cells on this heat map represent is the overlap. So we compute, using the hypergeometric test, the extent of overlap between the change in the mouse brain and what is seen in a given module, human co-expression module, and then that gives us a p-value that we shade in these cells. This allows us to visualize which mice are activating patterns of gene expression that we see in human brains. And with that, I'll take you through. So here I am showing you data from the 54 different mouse Alzheimer's disease model expression signatures. So this is data from 12 different mouse models, and we've heard about some of these models. These include amyloid precursor proteins transgenic mice, shown in this group here, some tau or MAPT transgenic mice, and some miscellaneous models. And you can see the heat maps and the human modules or the rows as shown. So what can we take from this? I should say that, all mouse models of Alzheimer's disease are not the same. Even mouse models that are expressing the same human proteins. There are different levels, there are different tempos of progression, there are different time courses of synaptic changes, the development of pathology, neurodegeneration, cognitive impairment, and the like. So that's why we don't expect to see all models showing exactly the same behavior on these heat maps. So the first thing that you notice are these clusters. And you can see a group here that's activated across a large number of models, and a second cluster here that are activated across a large number of models. Now, it turns out these clusters are strongly enriched for gene sets that we associate with specific neuronal cell types, okay? So this group at the top involves genes that are expressed in microglia, or immune-mediating cells. This second cluster are genes that are expressed predominantly in neurons, including a lot of synaptic genes. Then there are astrocytes, oligodendrocytes, and some less well-defined gene sets. What you can see is that modules that are enriched in neuronal or microglial genes are strongly activated by many models, and those are these two bands at the top. Now, one thing you may be thinking, okay, there's overlap, but is it the same change in the mouse and in the human? It turns out that most of the overlaps are indeed concordant between human and mouse brains. And what you can see that here, now we've color-coded the overlaps, red where they're concordant up, blue for concordant down. Microglial modules are largely up, which goes along with inflammation accompanying Alzheimer's disease changes. Neuronal modules are mostly down, which goes along with neuronal loss and deleterious neuronal changes. Okay, so the first thing we looked at is, are there specific patterns that demonstrate tau or amyloid-based specificity? And there are some, but not many. For example, we rarely see co-activation in a single model of both microglial and neuronal expression signatures. But we see that more commonly amongst models that are either tau transgenic models or are known to develop robust tau pathology. So this may be a relatively specific signature of tau-related neuronal injury. In contrast, I'm highlighting here some changes in oligodendrocyte-enriched modules that are activated predominantly, noted with these arrows, of APP transgenic mice, which we don't see down here in any of the tau mice. So they're absent over here, but present over here. The next thing we wanted to look at is age or dynamic changes over time. Of course, the great thing with a mouse is that you can take a specific mouse, age it, sacrifice it at different times, and look at the changes that are determined. You can't do that in humans, of course. And so what I'm showing you is that for these APP transgenic mice, we had several time points. What you can see is that initially the brain transcriptome is quiescent, but beginning at 6 months, microglial modules are strongly activated, and that is sustained up to 20 months. Oh, and I wanted to mention that we know that around 6 months from other studies, synaptic loss is occurring and neurodegeneration is beginning to occur. So there's concordance there. In tau mice, you can see at 2.5 months, there's no microglial activation, but there are changes in neuronal modules that then flips. And now we see activation beginning at 4.5 months of some of these inflammatory changes. So we can dissect these age-dependent differences. We actually developed a rating scale for the pathology that's been published in each of these different models. We use these green shading to indicate level of pathology and just look for global changes in activations and overlaps in relation to pathologic burden. And it fits with what I've been suggesting earlier, that early on, we tend to see activation and overlaps of neuronal modules, whereas later, as pathology is strongly developed, we see activation of these microglia gene-enriched modules. Okay, this is the other slide that's really important, because it was one of, I think, the surprises of our study. We looked at a lot more than Alzheimer's disease mice. There were a number of mice that were just pure aging mice, in which mice had been aged out, and we could compare the transcriptome in aged mice to young mice. And surprisingly, these microglial and neuronal modules are strongly enriched in aged animals, which suggests that these signatures are not terribly specific for Alzheimer's disease pathophysiology. So, for example, this particular mouse expression pattern is from hippocampus tissue in a 24-month-old mouse versus a 3-month-old control. Others have seen some results consistent with this. Moreover, when we look at mouse models of a whole variety of neurodegenerative diseases, including Huntington's disease, FTD, spinocerebellar ataxia, prion disease, we see very robust activation of these microglial and neuronal modules that are found in Alzheimer's disease human brains. So again, these modules are not terribly specific to Alzheimer's disease, but there's an important paradox here that I can address in questions or over lunch, and that is that we do know from Alzheimer's disease genetics that these pathways involving microglia and inflammation cause Alzheimer's disease or affect Alzheimer's disease risk. But the transcriptome suggests that it's not terribly specific. Okay, so I'm just finishing up now. If you've been astute, what you've noticed is that there is this region of the heat map through here, which is largely quiescent across all the Alzheimer's disease mice. And in some ways, this is the dark matter of the Alzheimer's disease transcriptome. These are human brain expression networks that are significantly associated with Alzheimer's disease, but do not appear to be represented in our currently available mouse models of Alzheimer's disease. So in many ways, this could be why our mouse models are not working so well for us in drug development. And when you look at what these modules are involved in, they're involved in biological processes that we know are important and interesting from the standpoint of Alzheimer's disease, including mitochondrial biology, unfolded protein response, DNA repair pathways, which certainly relate to what Bess was telling us about. So we had the idea that if we can identify other mouse models out there that activate these pathways, they might be good mouse models of Alzheimer's disease. And so we think these are AD transcriptologs. These are mouse models of Alzheimer's disease at the level of the transcriptome. They could be out there, but we don't know it because someone wasn't trying to make a mouse model of Alzheimer's disease. So this large heat map shows all of the data that we processed as part of this project. And I then give you a magnification, indeed, of several mouse models highlighted with arrows here. You can see that activate overlaps with this dark matter region of the AD transcriptome. And I don't have time to go through them kind of in detail, but here's one interesting example. This here is a model in which somebody manipulated a hypothalamic gene that causes a hypermetabolic mouse and a lean phenotype. And that seems to activate a mitochondrial co-expression module that is not activated by any other Alzheimer's disease mouse. And the last thing I want to say is that everything I've been talking about is really up here in the pathway of Alzheimer's disease. It's really trying to understand the triggers and their effects on the transcriptome, up versus down. Of course, any change that is observed could be protective or amplifying for disease. Although I don't have time to tell you about that, what we're just completed doing is manipulating 350 genes prioritized from across these 30 modules in fruit fly models. And we're now integrating those results with similar transcriptomes from fruit fly brains to try to figure out which particular genes within networks are causally associated with age-related neurodegeneration. And with that, I'll just summarize what I told you. Our current Alzheimer's mouse models are predominantly activating brain responses that appear to be nonspecific. They overlap with aging and many other disease models. Second, there are certain expression signatures that may be able to differentiate tau versus amyloid precursor protein-based injury, but not many. I expected a lot more. Third, there are age-dependent expression changes, and so we can begin to dissect time-dependent changes. And we can use these types of transcriptome overlaps to possibly identify new, non-obvious models of Alzheimer's disease. And we are hopeful that fruit fly models can help us accelerate the pinpointing and mapping of causal hubs within these very large gene regulatory systems. And because I'm over time, I'll just let you all absorb the large and rich collaborative network that I've been lucky to work with. And the final point I'll say is that all the data I showed you is available in the public domain on this website. So ask me if you're interested, but it's open science. Thank you.
Thank you. We have time for questions. No questions?
So I'm far away from this field. My research, but I didn't get, maybe I missed it. So how does tau levels and spreading increase? What's the trigger? That didn't come across.
Yeah.
And then I have another question.
Okay. Yeah, what's the trigger of tau spread? Okay, so we know from the neuropathology that tau accumulates in certain areas of the brain at a very, very young age. Even between 10 and 20 years old, you get tau accumulation in the locus ceruleus, and that increases with age. I think people who are between 20 and 30, 80% of people have accumulation of tau there and no cognitive problems.
And is it transcriptional or
No,
post-transcriptional?
at the protein level. This is like disease-associated tau, actually, accumulation. But it's not in Alzheimer's disease, as we heard yesterday, there's not really a spread of tau from those age-associated areas where tau accumulates until you start to get the A-beta pathology as well. However, there are all these other non-Alzheimer's disease tauopathies where tau is accumulating, and it's wild-type tau for the most part. We know traumatic brain injury can induce tau aggregation. There are other stimulators like mTOR activation. There are a lot of things that can cause tau to accumulate.
Just a quick other question. We use hypomethylating agents in leukemia therapy, which leads to induction of human ERBs pathway and beta interferon and innate immunity getting activated, which is felt to be now the cause of or probably explaining the activity of these hypomethylating agents in myelodysplastic syndromes and leukemia. You're describing through, in this disease, the same kind of pathway occurring through PV and piRNA manipulations by the disease. So do you find parallels in the two that could be targeted?
Yes, so absolutely. This is kind of the basis of a collaboration between Jim's group at MD Anderson, that neuroscience group, and myself. Maybe you should talk about it, but it's basically looking, utilizing what we know from cancer and drugs that are effective in modulating the epigenome and seeing if we can apply those to tauopathy. Do you want to say any more about that?
No, that was it. There's a rich history of working in epigenetics for cancer, and we're just trying to apply it to Alzheimer's based on Bess's recent data.
Okay, thank you.
The last talk made me start to think about big data, data analytics, artificial intelligence. Are there efforts to look at data sets like what you were showing that go that direction?
Yeah, there absolutely are, both in genetics and the genomics of Alzheimer's disease. I think you're absolutely right. We're just beginning to scratch the surface of what's possible in these large data sets, I mean, including some of the types of data I showed you, but also other data that exists in the field. From these Alzheimer's disease brains that I'm talking about, this large AMP-AD consortium bank of data of more than 2,000 brains, beyond the RNA-seq data, there's now going to be comprehensive proteomics, unbiased proteomics, so the whole proteome. Also, there are epigenomes from many brains, including both histone acetylation, DNA methylation, and so we just have this multi-layered type of data. And I think those of us who are in this consortium and others who work with these data are, I just think, just beginning to scratch the surface of how to analyze it appropriately and extract meaning. And I think machine learning approaches and approaches that haven't yet been developed are going to be needed.
Thank you. Next question.
Well, my question is probably related to the first two talks. I think both of you mentioned about axonal injury and neuroinflammation. And I'm very much interested in this field, and what I want to know, when neuroinflammation happens, eventually you indicated the cell eventually die. But in between this process, is there any way to measure the communication, especially the action potential that is transmitted within the axon? Does the inflammation hamper or disrupt this communication?
Right, we're very interested in that question too. So that's a bit of a mystery in a way, that how the communication happens between neurons and microglia or Schwann cells in the periphery. Once their axons are injured, you see an immediate response by the immune system From my specific project, when we block DLK and prevent this axonal stress response, the microglia don't respond. So we think it's an intrinsic neuronal signal that's part of a biochemical cascade that's coming from the neurons, leading to the activation of the microglia, which, of course, is meant to be beneficial. But maybe in chronic disease states, it's not. And so the thinking is there may need to be pushing resolution of this inflammation in order to achieve, a more of a homeostatic state.
And the last question, over here.
Hello, my question may be specifically related to Dr. Jim Ray about the axon, maybe not the AD. So I'm a PhD student. My project is related to, my project is develop a new technology to open the blood-brain barrier for drug delivery into the brain. So our method works. We can open the blood-brain barrier, and then we checked the neural response after we open. So our results show the axons, the axon marker is at the 5 minutes after we open the blood-brain barrier, it's fine, it's normal, but at 6 hours, it's down-regulation, and then 12 hours later, it's come back. So is this good sign, or do we need to check other marker or something for the question? Because I'm not in this area, I'm not familiar with this area.
Oh, that's an important technology you're trying to develop. There's a huge need to get drugs across the blood-brain barrier, which is what you're trying to do, right?
Yeah.
Yes.
Could you say again? Sorry.
So you're trying to develop methods to get drugs across the blood-brain barrier.
Yes.
Yeah, that's the most advanced drugs for Alzheimer's disease are all antibodies, which are very poor at crossing the blood-brain barrier. So what you're doing is very important, and I'd be happy to discuss with you in more detail maybe some safety concerns that we can guard against.
Sure. Yeah, because we already demonstrate we can deliver the antibody, human antibody, across.
Then I definitely want to talk to you.
Yeah,
sure.
there's going to be time in a couple seconds at the break.
Let's thank this panel, George and team. Thank you all very much for doing a wonderful job.